Hey everyone, and welcome to Science in Real Time. This is the show where we sit down with the people pushing science forward and talk about what's really happening behind the breakthroughs. Before we dive in, make sure to subscribe so you never miss an episode. We've got some incredible conversations coming your way. All right, let's get into it. That it would be pretty fitting to start the podcast by hearing a little bit about who you folks are and welcoming you into the podcast. So why don't you share with our listeners, who are you and what are you here to talk about today?
My name is Magdalena and I'm Lead Molecular Biologist at Ardigen. And as it sounds, I have a biological background. I graduated in biotechnology, have PhD in molecular biology, but I have worked as cell biologist for all my professional life. I developed cell-based assays for small molecule screening, focusing on high content screening. And I started my journey with high content screening already from my postdoc at Aventis Pharma, which is now Sanofi in Paris. And I worked with the prototype of one of the first high content screening instruments that it's not produced anymore now. And then I moved to Italy, where I worked for a small biotech, set up the high content screening platform there, and then moved to Sweden. First joined AstraZeneca, also as a high content biologist, and then moved to Karolinska Institute when Astra closed the site that I used to work at, and also working with high content screening all the time. And around 6 years ago, I came back to Poland and was setting up the high content screening platform for Polish Academy of Sciences. But then Ardigen approached me, and then when I learned that they are developing the AI-based platform for analysis of high-content screening data, I was thinking, this is something that I was dreaming about for all my time that I was producing such data, and I didn't have such a tool. And I thought that would be cool to be a part of that, so I joined Ardigen.
I don't have such a long history of my professional experience as Magda. I studied computer science. My background is in deep learning, AI, and computer vision mostly. During my master's, I met a great advisor who I started working with analysis of images. And after studying computer science, I worked sometimes for a consulting company with boring business stuff. But after some time, my advisor actually told me that there is work at Ardigen and that they are focusing on computer vision for different types of biomedical imaging. And I joined at that point, and I started working first with histopathology images, and almost right after that with high content screening. And when the pandemic hit, I had a little bit too much of a free time. I started my PhD, which was like a joint PhD with company and the university. And it was mostly focused on high content screening, but also other types of microscopy images. I worked with light microscopy for bacteria and fluorescent for high content screening. I finished that some time ago, and during my time at Ardigen, I've been working with biomedical imaging, but also with software development, mostly with the PhenAID platform that I was a product owner for quite a few years now. And yeah, so I'm combining both the computer vision stuff, the software stuff, and learning about the biology along the way.
That sounds like a wonderful career track from both of you. And I think that now I would love to jump into talking a little bit about the origin of PhenAID. That's one of the tools in Ardigen that has been caught my attention, specifically because of the problem that it set out to solve. And I would love to go a little bit more in depth with you folks. And I figured that it would be good to start talking a little bit about what was the scientific or practical challenge that originally motivated the creation of a tool such as PhenAID.
I think, first of all, there was a lack of such a tool not only for the market, from the business perspective, but also generally. There are some open source tools, but nothing as PhenAID. So something that is user-friendly and biologists can use. So this was the first motivation. So, and also this what we've seen that our partners, our clients, and big pharmas, they are lacking such a tool. So something that can help biologists, but also we see from clients, PhenAID is replacing whole team of the data scientists. So there were those two motivations. So like first, very scientific, like to be able to extract all the informations that are encoded in images, extract them in an easy way, simple way. And the other business that we see that it's something that big pharmas are looking for and working with now.
Yeah, and it was also like a combination of few things, because at the same time we got a really big progress in computer vision and in AI models, a very big progress in the hardware. So GPUs, but also like a laboratory hardware, and the interest of the community. We were just before the jump cell painting consortium started, and all of that went really mainstream. So the combination of like everything really helped us with building such tool.
Phenotypic screening as a concept isn't necessarily new, but what PhenAID is doing, it's very much revolutionary. So I'm wondering what felt limiting or broken about existing approaches and how PhenAID is helping mitigate those.
Yeah, so you're right, because phenotypic screening is not, it's actually older than target-based screening. Yeah? So the simplest phenotypic screening is just like cell viability assay. Yeah, but it was for a long time was seen as a low throughput type of experiments and also not giving a lot of information. So the first turning point was the development, so introduction and development of high content screening. Over the last 2 decades, it's evolved enormously. RSLE instruments are so fast now, and they can produce really good quality of images. We have AI now and computer vision, development of computer vision technologies that can be applied to analyze those data. And now it's moving from the simple, very complicated experiment and a simple measurement to not as complicated, fast experiment that can give a lot of information. Researchers can look at cell morphology at scale, because you can see this morphology of thousand cells, of million cells actually. And now it's not anymore really black box as what before.
And with that, I want to jump into talking a little bit more about what PhenAID does exactly and how does it work compared to other tools that are out there. So for starters, I was thinking that we can talk a little bit about specifically for the researchers that are hearing about this for the first time. How would you explain PhenAID in a few sentences? What would you say PhenAID is basically?
Ardigen PhenAID is a technology platform, so we provide both services, but also an application that allows you to package everything and do it yourself. And we provide a platform that, first of all, ingests data, no matter the size. We can work with any size and also any type of data, meaning the morphology. And it helps you prepare the data for computer and for the GPU, and then provides predictions, generative processes, and at the end you can get biological insights from that. And we provide a lot of customizations for the platform, so actually you can work with the model that's best for the data you are using. And we are using both morphology, but we also add other omics, chemical structures, transcriptomics, proteomics, and so on.
What type of phenotypic or image data does PhenAID works best with today?
Most often work with clients' data, and this is because still high content screening has some batch issues, problems with like joining data from different sources. So we mostly work with like a single cell, single source data, but we do have methods for combining other cell lines and other sources and getting those batch effects and those issues. But it's still a problem that occurs in real life. So we mostly work with high content screening and with cell painting, because the availability of the data is right now the biggest, and we have both public data for that and also like proprietary data from our clients. But we can work with other protocols and other types of high content screening, even in some cases with Brightfield if someone wishes to.
Going back to the idea of PhenAID challenging this black box concept that we see in a lot of other tools, I'm wondering how PhenAID it differs from traditional imaging tools and challenges this black box AI model.
With PhenAID, we got much more than just image analysis or just an AI, because we do use some parts of like a classic image analysis. We provide, for example, quality control steps that can be sometimes done with pretty easy methods, and sometimes it is a black box AI. And we have AI models that are customly trained for the data. So you, first of all, you know that it will work with the data, and we always validate. So we don't say, oh, here is the model, we will give you the application, just run your data through it. We will validate it and show you that, yeah, it works. This is the performance of the model right now. We can retrain it in the future. And you will like know that for this essay, this is the performance I'm getting, and for this, it's even better. But also it packages everything into like a modular scalable platform where you can push even more data and like pick and choose what you want to do. And well, finally, the person in the researcher can do it themselves. They don't need us to run the models. They can do it themselves and have all this power. And even, as I said, retrain some of the models on their own if there is more data.
Considering that trust is a big concern when we have AI, how does PhenAID ensure its insights are biologically meaningful?
For biologists as me, the best way is to validate in a wet lab. So we as Ardigen, we don't have wet labs, so we cannot validate. We can only validate retrospectively, but we know from our clients that they validate our results in the laboratories, and it works because they still continue working with us. So that means it works. Unfortunately, we cannot disclose any confidential information, but I think this is the proof that it works, because they are applying the results of our outcome of our models to the drug discovery pipelines. So it works. Of course, it doesn't work for everything. They're not for all the cases. And you have to keep it in mind as well. So it's not like solution for everything, but it's really, really helpful in a lot of cases.
And I think that that's where being able to work directly with the clients comes really handy, and you folks can both support each other in a very synergic way. So I think that that's really, really powerful. And going a little bit deeper into the interpretability, I was wondering how important interpretability is when designing a platform of this nature, and what does it look like for users? So basically, what does the users receive? How are they able to interpret the data from their end?
Interpretability is like from the computer vision standpoint is solved in a lot of natural images, but for biology it's very tricky because, well, we can describe that something is happening on the image and like say, oh, the model is doing this prediction because it sees that something is happening in this channel. However, if we don't have a professional who will say that yes, this is true, we cannot validate the description or the explanation. And well, with natural images, we have a massive amount of data on the internet. Like you will get an image and description for like unmeasurable amounts of data. With high content screening, we don't have that. We have like a small percent of people who actually know what's happening on the image and not everything. So we've seen some cases where we can say that. And we have some examples where we can say that, oh, we see that the change is happening in the specific cells on the image, but not always you can actually say what's happening. So with this in mind, we don't provide like the exact descriptions of what's happening, but we will give you like an introduction to interpretability of saying that there is a probability that this is happening, and you can like based on that make like believe the model or not. And we also train the model in a way where it will predict something if it's more sure of it, and not like, oh, I think it's 50-50 that it's this label or something. So yeah, interpretability is very important, but it's still very tricky because you cannot validate a lot of descriptions.
It's another layer on that, because it has to be really these results of the AI they must be put in a context, in biological context. And it depends what is your biological question. You can interpret the same outcome different way. So it depends if the effect that we predict is if it's something that you are looking for, or is it the adverse effect in your case. So it really requires a lot of deep biological knowledge in exactly this biological topic, and we cannot provide everything like that. So it's also, we don't think it's worth to put an effort for that, because you cannot generalize it. It's better to provide something like a solution that then the biologist that has this deep knowledge can interpret.
I couldn't help but start thinking about what lies ahead. And I'm wondering what excites you most about the future of phenotypic AI right now, with all of the new changes and all of the new technologies that are coming out.
So with mostly new technology and that we are getting bigger with everything. So we get more resources when it comes to computing power, and we get more resources when it comes to lab hardware. So I think we are going in the direction where we will be doing a lot more experiments and a lot of more screening, and we will go into the direction of more general models, more general AI models, where we won't be limited to one type of problem or one cell line. Maybe we will be at some point able to predict from one model that can do it all. There are some works right now, for example, Juan Saicedo Lab is doing, they recently published a paper where they are using 75 different datasets for model screening. So yeah, I think we will be going in this direction where the resources will be used, the data will be there, and maybe we'll even get some more descriptions and more, labels for the data and more trustworthy data.
And from my perspective, what really excites me is a virtual cell. It's somehow connected to what Ada said. So she sees it from the data scientist perspective, but I see from biology perspective. Of course, I love to be in a lab, but if I could run my preliminary experiments in silico and then go to the lab only to validate them, that will also save a lot of money, a lot of time, and speed up everything. So it's also something that it's not that new, but now it's getting more and more attention. And I think this is the future, the virtual cell. Maybe one day we will be able to test compounds first in silico.
As a wrap up and a call to action for this interview, I wanted to bring up what is one misconception about AI in biology that you wish that we would challenge and potentially go away when it comes to AI?
I think scientists, they want to they want that AI confirms what they see by eye and what they assume, or what confirms their hypothesis. And we should look at the AI as something that is discovering something that we cannot see as a human. So this is the misconception. But of course, I also understand that scientists must trust in what AI is producing. But from the other hand, AI should understand that AI can make mistakes as humans do. So why we accept mistakes done by people and not by AI? But then algorithms can analyze much more data than human can. So even if they make the same number of mistakes or the same amount of mistakes as the human, finally they will produce much more correct data. This is something that I think people should consider. Scientists, they are skeptical to use of AI, should think about.
Yeah, totally agree. We had some cases where we were talking to the client and they were like, yeah, but why does it say it? I'm not seeing this thing that's predicting. Why does it do it? And it's like, well, AI is something different than a human eye.
I'm wondering, as a follow-up, what advice would you give researchers who are curious about AI but maybe hesitant about adopting it, or even hesitant about exposing themselves to it? Is there any advice that you would give to them?
Yeah, I would say there's nothing wrong with trying at least once. And we're at the times where it's not an option anymore, because at some point, at some part of your life, even if it's not the lab part and the scientific part, you will have to interact. So maybe let's get to know the AI and those models in any setting, because this knowledge will transfer to all the other types. But yeah, it's like it's the newest thing and it's happening, so there is like no choice.
Yeah, just learn how to use it and how to make profit out of it.
So, with that, I wanted to wrap up by naturally doing a call to action. I was wondering, where can folks connect with Ardigen, yourselves, and be able to learn more about PhenAID if they wanted to use that technology?
Yeah, you can always visit our website. There are some information on the website, ardigen.com, of course. And then you can connect with us also via web or via our social medias as well. And then we are happy to talk to any people that want to talk to us and explain more details. We are also attending the conferences, and our main conference is SBI2. So for years we attend the Boston conference, but I also want to advertise because I'm in charge of organizing European SBA2 meeting in Cambridge in July. So I invite everybody.
Yeah, we are primarily based in Poland. However, we do have presence in States and the rest of the Europe. So if you want to meet in person, we do have people that are close by, but we are always open to have a call and discuss any of those topics.
Absolutely. And we will have all of those links in the description box below for all of our listeners to be able to very easily access, including the conferences that we have been speaking about, so that you folks can actually go and meet the Ardigen team in person if you wish. And with this, we conclude the interview itself. I wanted to thank you so much for joining us and for sharing how PhenAID is really helping scientists see biology in new ways. I think this is incredibly timely. It's an incredibly amazing technology. And for listeners interested in learning more, as I mentioned, we're going to have all of those links available for you. And yeah, this was amazing. Thank you so much, folks. It was a pleasure to have you here.
Yeah, thank you for having us and letting us speak about it.
Yeah, thank you very much. It was really a pleasure.
Thank you so much for tuning into Science in Real Time. We're really glad that you were able to spend time with us. If today's conversation sparked questions or ideas, we'd love for you to keep it going. You can connect with our guest and the Science in Real Time team using the links in the description box below. And if you enjoyed this episode, don't forget to like, subscribe, and share with someone who loves science as much as you do. Until next time, thanks for watching.